Hey, President Marion, in introducing the speakers, I want to just be clear from the start that just under 10 years ago, I was diagnosed with Parkinson's disease. So I have a very strong interest in what the speakers are about to tell us. The speakers have agreed to respond to questions. And if you can post your questions on the chat function on the Zoom, they'll be called up by Chris Stillwell as MC after the speakers are finished. The speakers are Clare Parish and Lachlan Thompson. Clare is a developmental neuroscientist with 20 years of experience in Parkinson's disease research. She has specialised expertise in the directional differentiation of pluripotent stem cells into various neural and neurological lineages, work that has contributed to the development of highly standardised protocols and the implementation of strategies to facilitate cross-continental research. She has utilised these cells and protocols to model neural development and disease and advanced cell-based therapies for neural repair, predominantly around the central theme of dopamine biology and Parkinson's disease. She has an international reputation in improving the safety and functional integration of stem cell-derived neural transplants for Parkinson's disease. Most recently, this has included successful efforts to combine both cell and gene therapy. Lachlan is a neurobiologist specialising in neural transplantation. He undertook his training in this area at Lund University in Sweden, where he specialised in transplantation of dopamine neurons in animal models of Parkinson's disease, including some of the first studies to utilise transgenic cell lines expressing fluorescent proteins in order to study the integration of implanted cells. Now based at the Florey Institute of Neuroscience and Mental Health in Melbourne, his group continues to work in the area of neural transplantation, both as a therapeutic approach for brain and spinal cord repair, but also as a way to understand the properties of neurons generated from human stem cells. Dr. Thompson is a board member of the Network of European CNS Transplantation and Repair, and the inaugural chair of the Asia Pacific Association for Neural Transplantation and Repair. Now over to Clare and Lachlan.
Thank you. Thanks for having us. Can I first start by apologising for supplying you with such long-winded introductions to read out, for which you did an exemplary job. So thank you very much for that. Thanks for having us today. Special thanks to Chris Stillwell, who we've been corresponding with by email, and also to Miriam Dexter and Ian Allen, who are also guests today that helped facilitate this. We would have loved to have been there with you all in person, but on the bright side, at least technology still allows us to connect. So that's the positive spin, of course. I'm just going to try and share my screen with you all. So Clare and I work closely together at the Florey Institute of Neuroscience and Mental Health, and we're just going to give you a short talk today about some of our work in the area of brain repair and using stem cells for brain repair. I'm sure we've all heard a lot about stem cells. They've come to assume this kind of magical property in the media as a kind of cure-all, and while no doubt there is really significant potential there, it takes a lot of hard work to realise that potential in a directed, meaningful way. And Claire's going to talk to you a little bit about using stem cells to make therapeutic cell types in the laboratory. And I'm just going to give a brief introduction to that by talking about one of the areas that's probably most promising for application of stem cells, and that's in Parkinson's disease, and really pleasing that there's a unique interest in that here today. So just to remind you of Parkinson's disease and how it's present in the community. So it's the most second common neurodegenerative disease, with only Alzheimer's and dementia in front of it. There's around 70,000 people approximately in Australia at any one time with Parkinson's disease. And the total cost to the community is around $10 billion per year. So although we all know about Parkinson's disease, but it's not necessarily front of mind all the time, and it is an enormous issue for the community, and one that will get worse with an aging population. So in terms of what it is, the cause of Parkinson's disease is actually the loss of a type of cell in the brain called a dopamine neuron. So you can see here, this is a tissue section from a normal person, and this black area represents the dopamine neurons. And in Parkinson's disease, there's a dramatic loss of this cell type. And these cells form a pathway that's really critical for motor function. So as you lose these cells, there's a progressive decline in motor function. And you can see just maybe at the bottom corner of my slide here, some old file footage from a clinic in Sweden of a patient with Parkinson's disease, and quite advanced Parkinson's disease. So you can see these classical features like tremor, difficulty with movements. So a big breakthrough in treatment from Parkinson's disease came in the 1960s in the form of a drug. It's called L-Dopa. Basically, it's a precursor for what these dopamine neurons make, which is dopamine. So patients that take L-Dopa have that converted to dopamine in the brain, not by the normal cells, these dopamine neurons, but other cells in the brain. And it restores this dopaminergic transmission that's important for motor function. And it's incredibly effective, and particularly in the first stages of the disease. You can see in this second little movie, the same patient after taking L-Dopa. So quite a remarkable single molecule that has a really significant impact on this motor dysfunction. And there's a film that some of you may be aware of called Awakenings. It has Robert De Niro and Robin Williams in it. It's a fantastic movie, aside from this interest in medical research, but it captures how a single molecule and its discovery can have such an impact on medicine. If you get the chance, it's really worth watching. The problem with L-Dopa, which is still one of our frontline therapies for Parkinson's disease now 60 years on, is that with chronic use, there's the appearance of side effects. And just in this final video, you can see a patient that while he has fluidity of movement, he has difficulties controlling movement, and that's called dyskinesia. That's a really common and still persistent problem with what are still our frontline therapies for Parkinson's disease. So there's absolutely an ongoing and urgent need for new therapies. And what I want to talk to you about a little bit today is the idea that we can replace dopamine neurons in the brain, not with a single molecule in a non-physiological way that then presents these side effects, but actually replace dopamine neurons in a true sense. And it's a therapy that was first developed in preclinical research in Sweden in the 1970s by this guy here called Anders Björklund. Now, here's my former mentor when I was training in Sweden in this area. This is just a picture of a rodent brain. And you can see back in the midbrain here, that's where the dopamine neurons normally reside. And they send these long distance connections up to the forebrain. So that's that motor pathway I was talking about that's really important for motor function. And you can see if you take a section through the forebrain, this dark area here represents a really dense pattern of that connectivity that's important for motor function. Now we can model what Parkinson's disease looks like in rodents by injecting a toxin into the midbrain, essentially kills those dopamine neurons in a very selective way. And you can see here in this slice through the forebrain that pattern of connectivity is gone. And those rodents will have, a mild motor dysfunction like we see in Parkinson's disease. And this key breakthrough came in the 1970s by Anders and his team when they discovered that if you take dopamine neurons from around the time they're being developed in the brain, from fetal tissue, that they can survive transplantation into a new environment. So this is just a picture of a fetal brain, and this area VM is where the dopamine neurons are normally generated. And when they're really young and immature, you can actually take them and place them into a new environment. And you can see here a picture of a dopamine neuron graft in a rodent. And this dark area is where it's extended out and is establishing new connections. And these experiments back in the 70s were so successful that they led to clinical trials throughout the 1980s in patients using human fetal tissue from elective abortion. And interestingly, the first patients to actually receive this treatment weren't Parkinson's disease patients per se, but were heroin users in the San Francisco area in the 1980s. So these patients started turning up in emergency room in San Francisco with this Parkinsonian-like state. So essentially frozen, having very little movement. And it was discovered that part of the production process to make heroin incorporated a toxic compound that kills dopamine neurons. And it's still a compound we use today to model Parkinson in rodents. So this is a picture of 6 patients that turned up unexpectedly in emergency rooms. And a guy by the name of Bill Langston, who was a neurologist at the time, kind of nutted out what was going on and that they'd ended up with this Parkinsonian state. And he was on a bus one day at a conference on Parkinson's disease with Anders Bjorklund, and they got to talking and Bill said, listen, you know, I've got these people turning up in my clinic. They look like Parkinson's patients and they have a loss of dopamine neurons. And Anders said, well, you know, we've been working on this therapy in Sweden to replace dopamine neurons. So, you know, I just wanted to highlight this as one of those everyday kind of events that, you know, underpins a scientific breakthrough, and that's how it happens sometimes. So 3 of these patients actually flew over to Sweden and had the procedure where they had new dopamine neurons implanted. And for any avid readers out there, if you want a really good read on this topic, there's a book called The Case of the Frozen Addicts, and it's a brilliant read about how, you know, serendipity and everyday circumstances can lead to medical breakthroughs. So I just want to show you some clinical footage of what this therapy looked like. So this is from a colleague in Canada, Dr. Ivan Mendes. Claire and I are not clinicians, we're research scientists, but there are some really talented clinicians working in this area. And this is one of his patients. This is off drugs, so he hasn't received any of his L-Dopa medication. And you can see very conspicuously the difficulty he has in performing basic motor tasks. And this is before any intervention. So you can see that tremor that, you know, most of us understand occurs in Parkinson's disease, and slowness of movement. And then a shuffling style gait, so a hunch posture, tremor again. And then this is particularly typical for Parkinson's disease at the top of the corridor there, as real inertia initiating a turn. And then you can see as he walks away from us, that shuffling gait and hunch posture. So he was a candidate for this therapy. This is now 3 years after he received a fetal tissue graft. I think it was from 3 or 4 fetal tissue donors, which is an issue, right? That's something that I'll come to in the next slide. And this is again off drugs. And this is an example of how this therapy can work when it works really well. And from a single surgical intervention and then sustained benefit, you know, in the best performing patients, 10 and 15 years and beyond, and they're patients that can come off drug therapy and be free of those side effects. So just to quickly summarize, you know, we have proof of principle that you can replace dopamine neurons in the adult brain, which is remarkable. There are benefits over conventional therapies. We need new therapies, and we understand how this works. The problem, of course, is that fetal tissue is not a suitable cell source. Besides being insufficient as a resource, it's impossible to standardize, and it doesn't always work as well as we just saw with that particular patient. So what about stem cells? And there's been a lot of expectation that stem cells may come in now and replace fetal tissue and allow us to push this therapy forward. And at this point, I just want to introduce my colleague, Claire, and give you some insight into another bit of serendipity, I guess, in that during my postdoctoral training with Anders Björklund in Lund, down here in the bottom of Sweden, Claire was actually working in the field of stem cell biology at the same time in Stockholm, which we didn't know at the time, right? This is complete coincidence. And then we happened to repatriate back to Melbourne around the same time in the early 2000s. And we both had a real passion to push this research forward. And our mission now is to develop a stem cell-based therapy as a cell replacement therapy for Parkinson's disease. And we're working closely together to do that. So I'm just going to hand over to Claire, if you're ready, Claire, to share your screen. And we can hear a little bit about where stem cells lie in this landscape.
Let's see how we go. Thanks, Lachlan. So I think I'm not going to give any sort of introduction, but just to keep the flow running, I think Lachlan's very nicely described to us the potential of this therapy, but also highlighted the limitation we currently have with working with tissue that's been obtained from human fetuses. So this is really looking at moving forward at what the alternative donor cell source can be. And of course, we're talking about stem cells. And I recognize this is a very broad audience here. And so I thought I'd just touch on what is a stem cell. And we define a stem cell as a cell that can self-renew, so it can divide to give rise to more cells that are the same. So one cell gives rise to 2 cells, to 4 cells, to 8 cells, 16 cells, and so on. And these cells, under the right instructions, can become cells with very specialized functions. So we can make a stem cell into a cardiac cell to repair parts of the heart, such as a heart valve, or in the context of Parkinson's disease, we can instruct these stem cells to make new dopamine neurons. So within our work in the lab, Lachlan and I are particularly interested in a class of stem cells that are called pluripotent stem cells. And the reason we're so interested in these cells is these cells have the potential, or they have unlimited capacity of self-renewal, and they have the capacity to become any cell type in the human body. So there's 2 different sources of these pluripotent stem cells. And the first one is what we call an embryonic stem cell. So this is where egg meets sperm. About 5 days later, we end up with this ball of cells called a blastocyst. And these are obtained from IVF clinics under consent by the donors. These are destined for destruction or donated to research, such as ourselves. And within this blastocyst, we can collect a small cluster of cells. This is what we call the inner cell mass. And we can grow these in a dish in the laboratory. And this is an example of these cells around about a week after we've collected them. And in the background, what you can see here is these human cells that have been growing for around about 20 years now. So this is a stem cell line that was first obtained in 1998 and has been ongoing growing through a number of labs around the world. The alternative pluripotent stem cell is more recently discovered, and this is by a Japanese group in 2007. And what they discovered is you could take a biopsy from an individual. This could either be a skin biopsy or it could be a blood sample. And these cells could then be what we call reprogrammed, where we introduce new genes into these cells. So we revert them back to this most primitive form, so that they're now almost like a naive or equivalent to our embryonic stem cell. Because of this, we can now instruct these cells to become all the various cell types, depending on what we're trying to achieve. And so you can start to understand now that you could essentially take a patient, such as a Parkinson's patient, take a skin biopsy, generate their own stem cells and implant them back into the individual. And this was actually done for the very first time just last year, where an American individual had his own stem cells created and transplanted back into his brain. So this has created real excitement in the regenerative medicine community, not just for Parkinson's disease, but for a plethora of different conditions, such as, as I mentioned, replacing heart valves or generating insulin-producing cells for diabetes. But how do we go about making this stem cell into a specific cell type? How do we go from this most primitive stem cell into the dopamine neuron? And surprisingly, this has taken us much longer than we anticipated. It was only in 2011 that the field managed to do this for the first time. And how we do this is a very simplified diagram here where we have the cells growing over a period of time in the dish, where we expose them to various cues. So in orange, maybe a series of cues to tell the cell to become a neuron or a neural cell, and then very specific cues to tell it which region of the brain we want these cells to become. In the dish, we can then stain these cells with different markers to confirm that we've made the right cell type. So this combination of red, green, red, and blue that we see here indicates that yes, we have in fact made a young dopamine cell, and this would be a cell that would be suitable to implant back into the brain. We can also mature these cells much longer. So this is some cells at about 80 days in culture where we can see they make very elaborate connections. And this can be really useful for us to better study a disease or to also look at developing new drug therapies. But in the context of transplantation, as we're talking about here today, we've been able to demonstrate that these pluripotent stem cell derived cells can actually integrate into the brain. So this is an example of 4 separate brains, and these are transplants made into the rat. And we can see that over the course of around about 6 months, we get these large transplants that can integrate or send connections throughout this host piece of brain tissue. So the brown staining here indicates the replacement of the dopamine connectivity. So in an ideal scenario, we have our stem cell, we make them into a neural stem cell or a brain-related stem cell, and then we end up with the dopamine cells that would be suitable to go into the brain. In reality, what we really have is an incomplete process that happens. So we'll have our stem cells, we start to differentiate them, but some of these stem cells reside in our cultures. As we move forward, the green cells, our dopamine cells remain, but we've still got this population of other cells present. And this can be a great risk when we look at going into the brain. We've got a highly proliferating or a highly dividing population of cells. And the risk could be that we'll generate a tumor, as you can see here. So to address this, one of the strategies we can use as scientists is to isolate the cell of interest. So in this regard would be to mature our cells and then collect just the green population of cells to implant just this select cell type into the brain. And we sort of refer to this as sorting the wheat from the chaff, or picking the green Smarties out of the box, as I may say to my 5-year-old daughter. So how do we go about doing this? We typically put a tag on the cell of interest. So most commonly used by scientists is a green fluorescent protein, and this is a protein that's extracted from the jellyfish. So we reintroduce this protein into our stem cells, and it enables us to track this population. So now what we're looking at is our cells that we've grown in the dish again. This time round, about 70% of our cells have this green protein expressed. So the process would be a dish of cells, mature them in the dish, and now we're going to put them through a machine where we sort the different green from the non-green cells. So we want to do a comparison about how much this really improves things. So we would take a population of unsorted cells, just the green cells, and all the non-green cells. And you can very strikingly see 3 different examples of these populations. This is what the typical transplant would look like. Brown staining again of the dopamine cells and their connections through the host tissue. And what you can observe is there's areas where there's a lot of dopamine cells present, but there's also areas that are very few in the number of dopamine cells present. By comparison, when we collect this GFP, or the green protein expressing cells, you can see that the graft is much smaller in size, but is much denser in the number of dopamine cells it has. And then conversely, if we take the non-green cells, you can see this a large amount of cell growth inside this transplant, and there's very few dopamine cells present. So this graft we see here is enriched with the dopamine cells, and in an animal model, we can prove that we can reverse all those movement challenges that Lachlan described to us earlier. So an alternative strategy to sorting the cells before we put them into the brain is to try and eliminate the unwanted cells after we transplant them. And we can do this by introducing a kill switch into our stem cells before we graft them. And without getting too technical, we essentially put in what we call a suicide gene or a kill switch into the cell. This is inside our stem cell. And then we can administer to the animals, and this is actually a drug used for cold sore, herpes simplex virus. We can administer a drug that's non-toxic, but when it's presented to a cell that contains this gene, it causes the cell to undergo death, but only when that cell's dividing. So as a consequence, within a transplant, we can selectively eliminate this dividing population of cells that may be of risk. So here's a typical transplant, which we can see the black staining here. And by activating this kill switch within the stem cells, we can make a much smaller graft, but this doesn't affect our dopamine functional population of cells. So that's strategies that look at improving the cell graft in its composition. An additional thing we can do as biologists is looking at also improving the environment into which we put these transplants. So we typically grow these cells in a dish. They're exposed to a cocktail of different nutrients. This is reminiscent of what the developing brain, human brain, looks like. Of course, these transplants are going into the aging brain, and they don't have many of these nutrients present. So we can strive to try and reintroduce some of those nutrients back into the brain. This can either be done by infusing proteins into the brain, which is quite a cumbersome process to do because proteins don't stay in the area for very long, or we can use a virus which we introduce into the brain. Not all viruses are bad. They've got some bad press at the moment, that's for sure. But we can introduce a virus that can make the cells within the brain create the protein we need. And in this regard, we've got a protein called GDNF, which is known to promote the survival and connectivity of dopamine cells. So we put our cells, our stem cells into the brain, we introduce this virus, which will enable the brain to create this protein, and this will cause the graft to survive better and make better connections. And here's just some examples. Here's the Lachlan showed us earlier, the Parkinson's model that we use in the lab. Here we are with a transplant, and a transplant that's now exposed to this protein. And when we look at various areas in the brain, you can see that there's many more, this brown staining indicates that there's many more connections being created. Just the very last concept I want to present you with is also some tissue engineering. So we also work with some engineers, materials engineers, that look to create scaffolds that can better support neural transplants. Here's an example of a gel that we work with. So this is a liquid when we have it at 4 degrees or at fridge temperature when we store it on ice. When we inject it into the brain, it forms this semi-solid gel structure. And when we look under the microscope at high magnification, we can see that this gel has very fine fibers in it. They're very supportive of these nerve cells that we're putting in the brain. So we can mix our cells into this liquid together with various proteins of interest and use this as an example where we can improve the survival and connectivity of these transplants. So you can see that stem cells really have amazing potential moving forward, but we always like to draw attention or present a word of caution when we talk to a general audience such as yourselves, and that is the concept of stem cell tourism. So you can hop on the internet and very rapidly find any one of these sorts of blurbs or one of these advertisements that are sort of selling stem cell therapies within unregulated clinics. And I really say that if something sounds too good to be true, it probably is, and we need to be highly cautious of these snake oil salesmen. These are often not regulated trials or no evidence of efficacy, and they really prey on the desperate situation that an individual is often finds themselves in. They're highly costly, and not only are they often not going to have any benefit for an individual, but they also present some risks. So in saying that, I hope that what Lachlan and I have presented to you here is really that there is great potential for regulated stem cell therapies. The field is really moving very quickly at the moment, but we need to make sure that we come at this with careful evaluation, that this is under regulated clinical trials. If we move too quickly in this context, we run the risk of damaging what may be an amazing therapy for many to follow. And so really, where are we now? I think we've demonstrated to you that we understand what we need to do and how we need to go about it. We now have a cell source, or we have a material that is highly standardized, and we can address both the safety and the efficacy of these transplants. What we need now to move forward to the clinic as a team is really getting that regulatory approval from the TGA. We need to look at manufacturing capacity here within our local networks, and we need to validate the cell batches that we intend to take into the clinic. And of course, this comes with requirements for both personnel and equipment to move forward. And so I'm going to pass it back to Chris, and Lachlan and I are very happy to take any questions that people may have. Thank you.
Thank you very much. The first question I've got is from Bev Brock. Does the original cause that destroys the dopamine neurons come back and cause the destruction of the newly implanted dopamine cells?
Great question. It's something we hear often. There is now evidence that there is a degree of transmission of the disease into the transplant. However, these are very baby neurons. Parkinson's disease propagates over many, many decades. And so the new young cells that we're putting in often outlive a patient. So around about 10% of the cells will end up developing some early signs of Parkinson's disease, but we've now seen evidence that transplants can function for up to 25 years in patients.
Thank you. I've got another question from our past president, Peter Rogers, and Peter's asking, does the development at Monash of eye blastoids from skin cells help open more opportunity for fetal tissue grafts? I hope I've done that question justice.
All right, and when I was
Go for it, Claire.
I don't, to be honest, I don't think this really changes the game too much for us. An eye blastoid is really looking at recreating very early development. We know we already have identified the cell source that is capable of making the dopamine cell. The eye blastoid is likely to also make a dopamine cell, but I think we have a product already in hand that enables us to generate the donor material that we need.
Great, thank you very much, Claire. You were talking about the tourism stem cells, which I got the gist that was maybe had an American bent to it. Do you have tourism stem cell opportunities in Australia, or would you regulate that out?
It's relatively well regulated here, yes.
Yeah. Yep.
At least for Parkinson's disease. I mean, you can still get a stem cell transplant for osteoarthritis here in Melbourne, and they do show some benefit in individuals, but it's sort of raising that level of caution about always checking in with your clinicians before you embark on any of these therapies.
Yep. So there's a couple of questions that sort of have the same theme, that are really sort of asking about people they know that, or relatives that have Parkinson's disease, and you're talking about something that you're in the research stage. So what's the next stage of potentially patients getting the benefits of some of what you've been discussing with us? And if people are interested in exploring that further, where would they go to?
Yeah, so when will it be available? That's a really important question. So we're just seeing some of the first regulated clinical trials for this conducted in the US and in Japan. So in Japan, they've already grafted patients. So as we indicate in the presentation, this has been done with fetal tissue. We know it works, but in terms of stem cells as a product, that's just being executed now. So in the States and in Japan. And these are big, really well-funded consortiums. You know, it's taken a lot of money and resources to get to this point. But we're hoping, you know, that in Australia we can have a node here, if you like, for patients. The other thing to mention is that Claire and I guess, are kind of working on the generation 2.0 of the procedure. So this will go ahead in the US and Japan, and we hope to do something better.
And that sort of leads us on, Lachlan, thank you very much. To the next question is from Christina Boys. Where does your funding principally come from?
Yeah, that's also a good question. So it's not simple, and I'm sure those of you that have friends that are scientists will know this. It is a real challenge in Australia, and it's only gotten worse, I would say, in recent years. So the indirect consequence of that is Claire and I probably spend 4 or 5 months of the year writing grant applications rather than actually doing research, where it used to be sort of 2 or 3 months a year and you could apply to the main federal funding system, which is now broken, in my opinion. But now we have to be much more diverse in our strategy. So we try and engage with commercial entities, philanthropy, wherever we can, to keep the team going. So it's tough, but we're lucky, Claire and I, that over the last 10 years we've managed to stay in business.
Thank you. And let me say that there are lots and lots of messages in the chat function about how great your presentation has been. So Lachlan and Claire, thank you. Certainly hit the right note.
No, it's been a pleasure, a real pleasure.
Somebody was reading the paper during the weekend, and there was something in the paper about light therapy for Parkinson's disease. You've got any comments on that or awareness of that?
I'm dealing with a bit of light therapy at the moment, actually.
I'm not sure if it's, I didn't see the light therapy. We actually had Lachlan and myself had an article in the paper, I think it was Monday, that was on some of the work that I presented to you here. It's a news story that we've just published on the concept of the kill switch to eliminate the unwanted cells in the transplants. I'm not sure if that's what was being referred to here.
I think good to mention there are other developing experimental therapies. This is one that Claire and I are interested in that we think has real potential, but there are other therapies out there. Deep brain stimulation is relatively new. That's been really useful for Parkinson's patients. So.
Well, thank you very much. And again, I'm going to hand back to Keith for the formal vote of thanks. Keith, back to you.
Thank you, Chris. Thank you, Lachlan and Claire. It's a fantastic presentation that made very complex technology understandable by us lay people. So thank you very much.
Thanks. Normally
we would provide a gift to you of a pair of rotary socks, R100 socks, R100 representing this year is our 100th anniversary. But they're important socks because they come from a company called Wilderness Wear in Preston. And I believe they're the only people who make socks in Australia for Australians. But they'll be forwarded to you in due course. So thank you very much.